Hello,
Good afternoon.
hello.
Hello.
I'm audible, Rahul sir.
Yes, ma'am. Am I audible?
Yes, ma'am. Yes,
Yes.
ma'am. You are audible.
Okay, okay. Ovulation, ma'am. May we start our
Okay.
today's ABP program?
Okay, ma'am.
Okay, okay, ma'am. Please continue.
Good afternoon, everyone. Welcome to day 4 of the Faculty Development Program on recent trends in renewable energy, organized by the IQAC cell of Regent Education and Research Foundation group of institutions. I am Abhilasha Sao, Assistant Professor of Basic Science and Humanities Department, and with me is Mr. Sabyasachi Mukherjee, Assistant Professor of Mechanical Engineering Department. We are delighted to be your host for today's session. Attendance in all sessions is compulsory to qualify for the certification. Today, we have with us Dr. Tado Gyadi, a dynamic researcher and engineer specializing in sustainable bio-waste management and renewable energy technologies. Dr. Gyadi earned his bachelor's degree in civil engineering from Mahatma Gandhi University and completed his MTech in environmental science and engineering from NERIST. He was recently awarded a PhD in civil engineering from NERIST in 2025, focusing on integrated bio-waste management through anaerobic digestion and co-digestion strategies in rural settings. Currently, Dr. Gyadi is serving as a junior engineer in Public Health Engineering and Water Supply Department of Arunachal Pradesh, contributing significantly to water supply and sanitation projects in remote areas. His notable publications include a review on anaerobic digestion of rice-derived food waste and animal manure in Bioresource Technology 2024, and a comparative study on compost activators in waste and biomass valorization 2025. His research aligns with practical, scalable solutions for rural infrastructure and sustainable development. Today, Dr. Gyadi will be speaking on the topic bio-waste to energy, sustainable management of organic matter for renewable energy generation. Let's now welcome Dr. Tedo Gyadi to take over the session. Sir, please.
Okay. Thank you, ma'am, for giving me this opportunity. Good afternoon, all. My name is Dr. Tado Gyadi. Today, I'm here to present bio-waste to energy. I will discuss about bio-waste to energy, sustainable management of organic matter for renewable energy generation. Coming to introduction part, first of all, I would like to say my work is more focused on the rural area of Arunachal Pradesh, especially in Arunachal Pradesh. So similar region can apply whatever I have learned in this experiment. So as you can see, the figure one, solid waste management plant at Chimpu, such a beautiful place which is covered by a solid waste. So from this figure, we can see there's a first problem is we need a large area for dumping site. Then there is a problem of the soil contamination, water contamination by due to leachate formation from the solid waste dumping at the dumping site. And then there is a problem of the air pollution. So with increasing in population and urbanization, there is an increase in the solid waste accumulation. So as you can see, according to World Bank, by 2050, waste is projected to be 3.4 billion tons, and livestock waste is 3.8 billion tons annually. This is a huge quantity which should be reduced. And then there is a problem of the from solid waste management. From solid waste, there is a generation of the methane when organic biomass decompose anaerobically. then it generates a gas, biogas. So biogas constitute of methane, carbon dioxide, hydrogen sulfide, and then water vapor. But among this, main constituent of the biogas is methane. And as you can see, methane is almost 20 to 36 times more global warming potential than the carbon dioxide. Now coming to with regards to Arunachal Pradesh context, in Arunachal Pradesh also the rapid urbanization and population increase can be observed. So with this, there is a problem of the solid waste management, because in Arunachal Pradesh there is a lack of infrastructure, and because of the poor planning, we are facing waste management in Itanagar. So we will see in the coming slide. As you can see, figure 2, garbage are directly being dumped along the river, and there is open burning at Chimpu dumping site, and then there is a roadside waste dumping. Now, figure 3 is showing some of the huge public NGOs are carrying out cleanliness drive at Itanagar. So they are doing their part. So from our researcher part, we are trying to convert this organic biomass into biogas, which can be used for electricity generation. So we can see the figure 4. First of all, what we have to do, we have to separate the matter. We have to separate paper, glass, organic, and then plastic, and then this organic mass. So for example, you can see the second cycle of the figure, where from the farmer we get fruits. So these fruits is used in the restaurant, hospital, food factory, etc. So from here we get the food waste, and then this food waste is converted to biogas or compost by the compost machine, and this compost is used in the farmer. So this is how the cycle goes on. Now coming to figure 5. Now I will discuss about, because I'm more focusing on the organic biomass. So what exactly is organic biomass? So organic biomass is the biomass which can be decomposed by the microbes. So for example, we have food waste, animal manure, agriculture crops, municipal solid waste, industrial waste is there, and then there are other which are inorganic biomass. For example, we have plastic and glasses, which cannot be decomposed by the microbes. And if cannot be decomposed by the microbes, then we don't get the biogas, which is a renewable source of energy. Now coming to figure 6, I will discuss about various factors which affect anaerobic digestion of organic biomass. So we have pH, carbon nitrogen ratio, temperature, hydraulic retention time, organic loading rate. These all factors will be discussed in the coming slides. Now I will discuss about what exactly happens within the digester. So when we ate for example, in my case, I have ate my food waste into digester. Within the food waste, we have complex polymers such as protein, carbohydrate, lipid is there. So first phase is hydrolysis. So during hydrolysis, this complex polymers is converted into amino acid, sugars, and then highly fatty acids by the hydrolytic bacteria. For example, we have Clostridium, Proteus vulgaris is there, Peptococcus is there. So after hydrolysis, during acidogenesis, it is converted into intermediary products such as butyric, propionic, valeric acids. And during acetogenesis, it is further breakdown into acetic acid and hydrogen and carbon dioxide by the acetogenic bacteria. And during methanogenesis, this acetic acid and then hydrogen and carbon dioxide is converted into methane and carbon dioxide. So this is how carbon flow inside the anaerobic digester and bacteria involved in the different stages of anaerobic digestion. Now, during my study of the many research paper, I found some research gaps. First, limited real world application. So most of the anaerobic digestion which I found during literature review was control lab based, which is not suitable for the rural condition. And then there is a temperature variability was not also considered, because in rural area temperature variation, there is a seasonal temperature is there, diurnal temperature is there, but most of the cases which research has been done in the field of anaerobic digestion was controlled control temperature. And regarding the small scale system for rural use was under research because they were more focused on the medium and large scale. So during my experimental work in Neris, I have mainly focused on the small scale system for rural use. And then regarding the microbial response in real condition, so in many of the research, they have only considered the stable condition. But in rural areas, if we go, so because of the variability in the substrate, because of the variability in the temperature, pH change, and according to pH change, when pH change, then there is a direct impact on the activity of the microbes, so which leads to the less production of the biogas. So now figure 8 is showing the co-digestion of animal manure and rice waste. So why I'm doing this co-digestion, I will discuss this in coming slides. So what exactly is co-digestion? So co-digestion is the digestion of two or more substrate. So in my case, I have digested rice waste and animal manure. When it is co-digested, it produces biogas, which can be used for electricity, cooking, lightning, so which is a sustainable source of energy. And then after co-digestion, there is a we get nutrients digested, so it can be used for fertilizer. So figure 9 is graphical abstract. It is graphical abstract. This is from one of my published paper. So in here, I'm showing the critical factors in anaerobic digestion of rice waste and animal manure. As we have discussed, the co-digestion of rice waste and animal manure uh, during the operation time, there's many factors which impacts this anaerobic digestion. For example, we have operational parameters such as hydraulic retention time is there, carbon nitrogen ratio is there, organic loading rate is there, temperature is there, then inhibitory factors is there, then microbial activity. These all factors impacts the anaerobic digestion, and because of this, it optimizes the biogas production and system stability. Now, after literature review, what I have found is there is a three range of temperatures are there. We have psychrophilic, mesophilic, and thermophilic. Regarding the psychrophilic, it is less than 20 degrees Celsius. So in psychrophilic temperature range, the activity of the microbes are not that active. So because of which the production of the biogas is lesser. So by the literature review, I found that mesophilic and thermophilic range is the optimal range, which gives you the best biogas. And regarding the optimal pH range, it was found to be 5.5 to 8.5. So if the pH is less than 5, it inhibits the activity of the microbes. And if the pH is greater than 8, then it is toxic to methanogens. If it is toxic to methanogens, then there will be a lesser methane in the biogas. So we have to maintain this optimal range. And then regarding the carbon nitrogen ratio, the ideal range was found to be 20 to 30. So if carbon nitrogen ratio is higher, for example, let's say 40, then there's a problem of the volatile fatty acid buildup. So when volatile fatty acid buildup, then there's a problem of the pH, because pH gets less than 5, which is not good for the activity of the microbes. And if the carbon nitrogen ratio is less than 20, then there is a problem of the ammonia buildup. So because of the ammonia buildup, it increased the pH more than 8.5, so which is not good for methanogens. And now regarding the microbial communities, microbial collaboration is key to stable biogas production, because as we have discussed earlier, the activity of the microbes during the hydrolysis, during the acidogenesis, acetogenesis, methanogenesis, the activity of the microbes are very important. Their collaboration, their synchronization activities are very important. Regarding the substrate characteristic, biogas production is directly dependent on the substrate properties. So for example, in my case, I have food waste and animal manure. Regarding the food waste, in case of food waste, we have particularly rice. Rice is high in carbohydrate and it is low in nitrogen. So because of which the carbon nitrogen ratio is higher in case of rice, and it is higher than the 30. So which is not good for the methanogenesis. And then there is animal manure. where high nitrogen content is there, low carbohydrate. So because of which carbon nitrogen ratio is lesser in animal manure. So it is not good for the activity of the microbes. So these all are factors which impacts the anaerobic digestion to produce biogas. Now I will discuss three objectives which I have followed in my during my experiment in the Northeastern Regional Institute of Science and Technology, NERIS. So first, to analyze the composition and quantity of biogas from co-digestion of food waste and animal manure. Then objective two was to study the effect of carbon nitrogen ratio on the microbial population in the co-digestion of food waste and animal manure. And then objective three was to isolate and characterize of most dominating microorganism involved in the conversion process of food waste and animal manures. Substrate material collection. I have collected my kitchen waste from Neri sources, and cow manure was collected from the dairy farm in Nirjuli, and red and white rice. Red rice was collected from three districts of Arunachal Pradesh, is coming Kurumkume, Papumpari district, and white rice was collected from Nirjuli. And similarly, animal manure, pig, chicken, and cow manure were collected from Central Pig Breeding Farm in Karsingsa, the Central Hatchery and Dairy Farm in Nirjuli. This is the map of the study site. Kurumkume is coming, and then Papumpari. This is the photograph showing the kitchen waste, and then white rice, red rice 1, red rice 2. Then we have the figure 12, which is showing the photographs of pig manure, chicken manure, cow manure, and its source. Now coming to experimental setup and design. During my experimental work in Neris, initially I have set up 3 sets of 2-liter bioreactors. Later it was expanded to 4 sets. I will discuss this in details in further slides. Regarding the feedstock preparation, each mix was diluted with water to make 1 liter slurry, and reactor were flushed with nitrogen for 3 minutes to ensure anaerobic conditions. And regarding the gas measurement, I have used water displacement method to measure the biogas production. Table 1 is showing the feedstock preparation. See, in first 3 sets, I have taken 100 gram of kitchen waste, and then 4:1 ratio of the kitchen waste to cow manure, and then individual digestion of the cow manure, 100 gram. So in subsequent set, I have taken 200 gram of substrate, where I have maintained the ratio as 1:1, 1:1, 1:0, 1:1, 2:1, 3:1. Now coming to figure 13, it is the schematic diagram of lab scale experimental setup. So as we can see, the three neck bottom flush is there. From two necks, we have inserted 2 electrodes to monitor the pH and temperature. And from the middle one, we have connected a pipe to collect the gas generated by the anaerobic digestion of the organic matter. So when this gas is passed through the water reservoir, then there is a burette, gas burette is there. So this gas displaces the water inside the burette. So that displaced amount of water is equals to the volume of the biogas produced. And figure 14 is showing how I have measured the gas composition. So you can see we have the 2-liter digester where it is all sealed, and then there's a cannula pipe is there. So from this cannula pipe, we insert this pipe inside the gas analyzer as seen in the picture below. In gas analyzer pipe, we insert this cannula pipe and then operate this gas analyzer. Then we get the methane and carbon dioxide composition. And regarding the chemical analysis method, I have for pH, I followed the Rhodes 1982. And regarding the moisture content goes for carbon content, Walkley and Black method. For total nitrogen, Jaldal digestion method. Total solid ace content, volatile solid, I followed the US EPA 2001. For carbohydrate content, I followed the Entron method. For crude fat, Soxler extraction method. So figure 16 is showing how I have calculated the carbon content of my organic matter, and figure 17 is showing the nitrogen content, Zeldal method, and figure 18 is showing the how I calculated the carbohydrate, organic matter, S content, and volatile solid, phosphorus digestion and fat estimation. Now coming to microbial analysis. For isolation of dominating microorganism, I have followed the standard lab technique of the ANESA method. In sample processing, what I have done is I have done serial dilution up to 10 to the power minus 6, and then I have cultured my microbes on the nutrient agar media. Regarding the culturing condition, I have maintained 37 degrees Celsius for 24 to 48 hours to isolate the distinct bacterial colonies. And regarding the bacterial identification, after isolation of the bacterial colonies, I have identified it using 16S RNA gene sequencing. So by 16S RNA sequencing, we get the sequence, and to know which sequence it is, we use the BLAST. We use the BLAST and created phylogenetic tree using Mega 10. So figure 19 is showing the preparation of the nutrient agar media for culturing microbes. And then figure 19 is showing the preparation of nutrient agar media. Yes, and after that figure 20 is showing the serial dilution of the sample. As you can see, the figure E, first the concentration is very high. After dilution up to 10 to the power minus 6, its concentration decreases. So the effect of decreasing this concentration we will see in the coming slides. So as we can see in the figure 21, from the serial dilution, we can see the figure C, first photograph, which is a too numerous count. We cannot distinguish which bacterial colonies it is. And regarding the second and third, so it is distinct colonies, distinct bacterial colonies can be observed. So from these distinct colonies, I have taken some sample, and by using the strict plate method, and I have stick it on the petri plate and then incubated for 24 to 48 hours. So after incubation, we can see the figure 22. We get this photograph. We get this result, different colonies of the microbes. Now coming to results and discussion. Table 5.1 is showing the material characterization of kitchen waste and cow manure. So we can see the table 2, the pH of the kitchen waste cow manure is in the range of optimal range, that is 6.5 to 8.5. In between it is. So it is good for the activity of the microbes. And regarding the volatile solids, the kitchen waste is higher in comparison with the cow manure. And regarding the carbon content, kitchen waste is higher with respect to cow manure, and then nitrogen content of the kitchen waste was found to be lesser with respect to cow manure. So accordingly, carbon nitrogen ratio of the kitchen waste was found to be much higher than the cow manure. And then regarding the fat content, the kitchen waste was higher in comparison with the cow manure. So now we will discuss about this table 5.1 in briefly. So higher volatile solid to total solid in kitchen waste was found. So it means higher volatile solid means higher biodegradable organic matter is present in the kitchen waste. So if higher biodegradable kitchen waste is higher biodegradable organic matter if it is present here in kitchen waste, then it will produce more biogas in comparison with the cow manure. And we have also observed the balanced composition of the kitchen waste. Kitchen waste is mix of fats, protein, carbohydrate, which boosts the activity of the microbes, and accordingly it will boost the production of the biogas. And regarding the carbon nitrogen ratio, we have observed kitchen waste has a high carbon nitrogen ratio in comparison with the cow manure. So we can see the carbon nitrogen ratio of the kitchen waste is around 38.59 and cow manure is 13.6. So it is not in between the optimal range. Therefore, during my experimental work, I have followed the co-digestion, because by co-digesting this kitchen waste and cow manure, we maintain the optimal range of the carbon nitrogen ratio, that is 20 to 30. So because it helps balance nutrients and improve the biogas production. Now coming to daily biogas production for the first three sets, as I have discussed earlier, we have three sets. First, individual digestion of the kitchen waste and individual digestion of the cow manure, and then co-digestion of the kitchen waste and cow manure. So from figure 23 and figure 24, we can see the figure, as we can see the bar graph, figure C is the co-digestion of the kitchen waste and cow manure. So it is much higher in comparison with the individual digestion of the cow manure or kitchen waste. Uh, Now coming to biogas yield and energy potential of different substrate. So after finding the volume of the biogas yield, uh, I, I have uh, I have found out its energy output. So for example, in case of kitchen waste, 100 gram, I found biogas yield is around 0.2406. When I convert this into energy output, megajoule per 100 gram, it is found to be 5.29. With respect to per kg, it is 52.9. Similarly, for cow manure, 40, and then for co-digestion of the kitchen waste and cow manure, it was found 64.8 megajoule per kg. So when I compare this with the traditional source of the fuels in rural areas, for example, we have firewood and dung cakes and LPG. So we can see figure 25. The kitchen waste is also biogas production of the kitchen waste is also much higher in comparison with the firewood and dung cakes. And regarding the cow manure also, it is much higher than the firewood and dung cakes. And see the co-digestion of the kitchen waste and cow manure is much higher than the firewood and dung cakes, and it is much higher than the LPG also. Energy output is much higher. So it is preferable in the rural areas. Now we will discuss about the subsequent four sets, which we have discussed earlier. We now in four sets we have maintained the ratio of 1:1, 2:1, 3:1. So now see the figure 27 and figure 28. With respect to cumulative methane production, we can see the set 4, which is in the ratio of 3:1, giving us the best result in comparison with the other three sets. And with respect to biogas production, also set 4 is giving us the best result. And then set 1 is giving us the least value with respect to methane or biogas production in comparison with the other three sets. So these four sets, why I'm doing this is I'm looking at the carbon nitrogen ratio, effect of the carbon nitrogen ratio, because in 1:0, why it is producing less methane production, because in it only contains kitchen waste, so there is no inoculum, there's no microbes. So for example, in case of 1:1, we have microbes, we have the cow manure where inoculum is there, so which helps in the digestion and which leads to the production of biogas. So that's why we can see the set 1 is giving us the least value in comparison with the other three sets. Now I will discuss briefly about what I have found in the four sets. Peak biogas. So it was observed around 9 to 12 days, especially 3:1, which is kitchen waste to cow manure ratio, gives us the best result. And regarding the methane yield also, 3:1 gives us the best result in comparison with the other three sets. Why it is so? It is because of the balance of the carbon nitrogen ratio. Because of the balance of the carbon nitrogen ratio, it leads to the better microbial activity, and it leads to the production of the better biogas production. So as we have discussed in with regards to kitchen waste, 1:0, only kitchen waste is there. So due to lack of methanogens in kitchen waste, which slowed down the degradation, so which leads to the lesser production in the biogas or with respect to the methane also. And co-digestion with cow manure significantly improved biogas yield, as we have discussed earlier. So regarding the optimal pH, it was observed biogas production best at pH 6.8 to 7.5. And with respect to the temperature, so during my experimental work in Neris, I have not controlled the temperature. I have just monitored the temperature. So during my experimental work, the temperature was around ambient temperature, that was 20 to 30 degree. More than 60 to 80% of the region in Arunachal Pradesh is having this 20 to 30 degrees Celsius, except some of the regions such as Tawang, which is very cold. So there's a temperature very less, and then there's a Anini temperature is lesser. But in majority of the areas in Arunachal Pradesh, this experiment result can be utilized. Now coming to table 4, here I will discuss about physiochemical properties of food waste and animal manure from different locations of Arunachal Pradesh, India. Earlier I have just discussed about the kitchen waste and cow manure. Now I am going to discuss about physiochemical properties of the white rice, red rice 1, red rice 2, chicken manure, cow manure, pig manure. So in here also the pH can be, as we can see, pH is in the range of the optimal only 5.5 to 6.5.5 to 8.5. Regarding the volatile solid, the rice is more in comparison with the manure, chicken manure or cow manure or pig manure. Regarding the carbon content, can also be observed that food waste is having much higher than the animal manure, and nitrogen content is lesser in rice waste in comparison with the animal manure. So accordingly, carbon nitrogen ratio is higher in case of the white rice, red rice 1, red rice 2 in comparison with chicken manure, cow manure or pig manure. Now I will discuss briefly about the table. We have observed the pH value. Food waste was around 5.1 to 6.71. And regarding the animal manure, it was around 7.53 to 6.63. With respect to volatile solids, it was found higher in food waste, that is red rice, in comparison with the animal manure, which means um, more biogas production, because higher volatile solid means higher degradable organic matter is present here in comparison with the animal manure, so which will leads to the production of higher biogas production. Regarding the carbon nitrogen ratio, we have observed that chicken, pig, and cow manure is having the lesser carbon nitrogen ratio in comparison with the rice. And with respect to phosphorus content, pig manure was having highest phosphorus content. So phosphorus content which boosts microbial activity, and high fat content was observed in case of the chicken manure, and in case of red rice it was very less. Now coming to the molecular characterization of dominant isolates. So during after 16S RNA gene sequencing, I have found my strength, which I have tested for the most dominant microorganism. I found that Klebsiella pneumonia, E. coli, Bacillus anthracis, and Chrysobacterium species were found to be most dominant bacteria in the conversion process. Regarding this Klebsiella pneumonia, E. coli, Bacillus anthracis, and Chrysobacterium species, these four bacteria are facultative bacteria. So it only helps in the initial breakdown of complex organic. For example, in case of the hydrolysis and acidogenesis, only it helps. But and Klebsiella pneumonia was consistently appear across rice and animal manure. I will show you this in the coming slides under scoring its importance and enhancing methane yields. So how I have confirmed this bacteria is by using the blast and phylogenetic analysis of 16S RNA sequence. Figure 29. So from during my experimental work in Neris, what I found is I have performed my work in lamella chamber, within the lamella chamber, so where I have not maintained the strict anaerobes condition. So because of which I have only able to get the fast growing facultative bacteria, that is Klebsiella, E. coli, Bacillus, and then Chrysobacterium species. I didn't get the culture independent microbes. For example, we have the hydrolytic, Clostridium, syntrops, metanogens, etc. To get this, because these all microbes are very strictly anaerobes. So if there's a presence of the oxygen, we cannot detect it. So for detect it, we have to follow the culture independent, that is 16S next generation sequencing. So if we perform this 16S next generation sequencing, then only we will be able to get this metanogens. This is the figure showing the primary sequence of the bacterial isolate of C22 and R23 samples. This is the Sanger method for bacterial identification of bacterial isolate, forward and reverse sequence. This is the forward and reverse sequence of the R23. This is just a representation. Now coming to figure 33, it is the phylogenetic analysis of the 16S RNA sequence strand, Klebsiella pneumonia, R23, Klebsiella pneumonia. So by using mega 10, we get this phylogenetic analysis of the 16S RNA sequence. So this is the phylogenetic analysis of the C22. Now coming to table 5, dominating bacterial strand identified from the food waste and animal manure. As we can see, the sample which was extracted from the white rice, so strain was identified as Klebsiella pneumonia. And with respect to chicken and pig manure, E. coli was found to be most dominant. And regarding the chicken manure, Bacillus anthracis was found to be most dominant. With respect to red rice, Klebsiella pneumonia. Cow manure, Klebsiella pneumonia. With respect to cow and pig manure, Chrysobacterium species. So in all samples, we can observe that Klebsiella pneumonia was predominant. Now coming to the conclusion part, as, as we have discussed, the peak production occurred 4 to 5 days for the first 3 sets in case of the first 3 sets, and with respect to the subsequent 4 sets, we get the peak production in the range of 9 to 12 days. Why it is happening? Because in the initial digestion, there is a abundant amount of the degradable organic biomass is there. So after degradable organic biomass exhausts, then there is a complex matter which takes much time to degrade. So during the initial phase only we get the peak production of the biogas. Regarding the cumulative yield, we have seen co-digestion of the kitchen waste and cow manure produce the highest biogas. And with respect to optimal ratios, the 3:1 gives us the best result with respect to methane, or with respect to biogas production. 3:1 was the best, and least was the 1:0, where only kitchen waste was digested. It was least because we have already discussed, because there was a absence of the inoculum in the kitchen waste. And with respect to energy efficiency, we have seen that co-digestion gives us the best result. This is around 64.8 megajoule per kg, outperforming the LPG and traditional fuels. When we compare with the traditional fuels, biogas exhibited 3 to 6 times higher energy efficiency than firewood and dung cakes, highlighting its strong potential as a sustainable fuel in rural area in Arunachal Pradesh. With respect to pH and temperature, what I observed during my experimental work is, first pH is shifted from alkaline to acidic, then it comes back to basic. And regarding the temperature range, it was 20 to 30 degree. And with respect to decomposition potential, high volatile solid in food waste promoted effective biodegradation and methane production. And with respect to microbial activity, a diverse microbial community, as we have seen in the table 5, it was dominated by the Klebsiella pneumonia, E. coli, Bacillus anthracis, Chrysobacterium, which played a key role in the initial breakdown of the organic matter. So with respect to the culture-based method, only reveals the few section of the community of the microbes, that is facultative bacteria, because methanogens microbes are very anaerobes, which cannot be detected by the culture-based methods. So for that, we need to follow the culture-independent sequencing. Now coming to limitation, in my experimental work, I have followed only a short digestion period, that is 27 days, and small scale bioreactors were used, only 2 liter. So we can go for further higher, medium to large scale. So, and then there is a narrow feedstock range. I have only taken the food waste and animal manure. There are many other organic matter are there. And then regarding the unaddressed temperature control, I have not controlled the temperature, because in rural areas, what matters is affordability and simplicity, because of the lack of infrastructure. So in rural areas, so that's why I did not control. I have tried my experimental work as practical as possible, which could be directly which could be directly adopted in the rural areas. So we have seen the temperature effect of the temperature, that's 20 to 30 degree. Within 20 to 30 degree, the performance of the biogas production was good. So in 20 to 30 degree, it is good. In maximum of the rural areas in Arunachal Pradesh, we can directly adopt this result. We can implement this result in practical sense. And regarding the only fast-growing facultative bacteria were able to isolate it, as I have discussed earlier. I have able to get only the facultative bacteria. I couldn't able to get the strict anaerobes such as the Clostridia, Syntrops, Metanogens, because it needs direct culture methods, because it is very sensitive to the oxygen, presence of the oxygen. Now coming to future research directives, long-term stability is long investigate the long-term performance and sustainability of anaerobic co-digestion system to ensure stable biogas production over extended period. In my case, I have only maintained it 27 days. So regarding the scalability, so we can go for higher, medium to larger scale. Regarding the diverse feedstock utilization, we can explore more feedstock, more organic biomass, such as agricultural residues. There are many other biomass are there which could be taken as a substrate. So regarding the microbial enhancement, I have only maintained the culture. Uh, I have only maintained the culture method. So I couldn't able to get the culture independent one. So we can go for the next generation sequencing to develop and optimize microbial conceptia for enhance the methane production and overall digestion efficiency. Now coming to list of publication, I published two research paper in ACI journal, and third paper is in second revision. Thank you. Any questions?
It was a wonderful session. So now it's time for a question answer session. So if anyone have any question, they can write in the chat box, or they can ask. They can switch on their mics and they can ask.
Excuse me, sir. Sir, I am Sridip Ghosh. Am I audible?
Yes, audible.
Uh, Sir, actually, here you have described all those bacteria, all those things. Sir, actually, as we know that sustainable development or green energy or renewable energy is the major area of research nowadays, because the non-renewable is degrading to a great extent. So, sir, the thing is that this bacteria or this biomass or biogas, this bacteria, uh, can it be very much, how much percentage of electricity generation, or you can say different types of other work in commercial areas or household areas? So how much percentage it can contribute to our society, like that, sir? If
Okay.
you
Uh,
understood, sir, my question, sir?
yes, I understood. So, see, from my only 2 liter of the biodigester, I could able to get 64 megajoule, which is much more in comparison with the traditional fuels, such as the wood is there, the coal is there. So, in comparison with, it is very good. So, according to the research I have uh, I have studied, so we can reduce up to 20 to 40% of the energy dependency on the on our fossil fuel. So 20
Okay,
to 40% we can expect.
okay, sir. Okay, sir. Thank you, sir. Thank you, sir.
Thank you.
So anyone having any more question, they can ask. They can type in the chat box, or they can ask directly.
Hello, am I audible?
Yes, audible.
Yeah, okay. So actually, it is a good presentation. So my
Thank you, sir.
comment is that what is the practical utility and significance of the entire work? That is my first question. And my second concern is that what are the basic differences from technical viewpoint between aerobic and anaerobic digestions of waste products? So I have
Okay.
these two questions. Thank you.
Okay. Okay. Thank you. Thank you. Regarding the first questions, sir, as I have earlier discussed about my experimental work, I have tried to follow. Uh, so, for example, in my case of the experimental work, I have not maintained the temperature. I didn't control the temperature. It was just monitor within the range of the ambient temperature, that is 20 to 30 degree. So within that range, it was working fine. The biogas production was also good. With respect to methane content also it was good. So my this uh, regarding the pH also I didn't control. So my result of this experimental work can directly be implemented in rural areas. Similar reasons in hilly areas, there are many hilly areas are there where 20 to 30 degree temperature is there. So directly this result can be directly implemented. So we don't. So as I have discussed earlier, in rural areas, what we emphasize is simplicity and affordability, because there is a lackness of the infrastructure. They cannot control the temperature, which is very costly for them. So they try to see as practical as possible. So my experimental work regarding the co-digestion of the kitchen waste and cow manure gives the best result which suits their requirement. And with respect to aerobic and anaerobic, so regarding the biogas production, so it is only possible by the anaerobic digestion. So when anaerobic digestion, in the absence of the oxygen, when organic matter is decomposed, then only it produces biogas. It constitutes of the methane, carbon dioxide is there, and then methane is the combustible constituent. So when we digest it aerobically, then we cannot get the methane. We cannot get this biogas. So this biogas only can be obtained from the anaerobic digestion. So this when we obtain this biogas, then only this can be used for sustainable energy. For example, for heating, electricity, and then there is a digested composite are there which can be directly used for the uh, fertilizer. So it will make you independent from the chemical fertilizers, less dependent on the chemical fertilizers. So in this way it is helping in the sustainable management of the organic biomass. Thank you.
Thank you so much, sir. So anyone from the participant, if they have any question, they can ask. So I guess there is no more question. So thank you so much, sir. A certificate of appreciation is presented to Dr. Tado Gadi, sir, as
Thank you, sir.
it is shown in the screen. And as we conclude today's enlightening session, we extend our deepest gratitude to Dr. Tado Gadi for sharing his invaluable knowledge and research on sustainable bio-waste management and renewable energy. Your insights have provided us with a deeper understanding of how bio-waste can be viable resource of energy generation. And a warm thank you to all the participants for their active participation. And now we look forward to seeing you all tomorrow on 17th May for the final day of the FDP. The session will feature an expert talk by Dr. M.K. Loganathan on application of AI and ML in renewable energy from 12 p.m. onwards. Thank you all for once again and have a great evening ahead. And lastly, a feedback link will be shared in the chat box as well as the WhatsApp group. Kindly fill the form to get the certification. Thank you, everyone. Thank you, sir. Thank you. Goodbye.
Thank you, sir. Thank you, sir.